Okay, so let's go ahead and start here. Smooth and cardiac muscle. Basically, looking at smooth muscle, we're thinking about ways that it differs from skeletal muscle, which we've talked at length about already. Smooth muscle lacks striations, hence the term smooth. It's found in internal organs. It's found in all of our blood vessels. It's in the uterus. It's in the bladder. It's in the GI tract. It's pretty much everywhere we have hollow visceral organs. It's under involuntary control. So it's being controlled by the autonomic nervous system. So we talked at length about the autonomic nervous system. And this is one of the types of muscles that is controlled by that division. It has cells that are differently shaped. So they are not elongated the way that we see with skeletal muscle. You can kind of appreciate that here. The way that we see with cardiac muscle, also appreciate that at the bottom here. Rather, the cells of smooth muscle are smaller and they are spindle shaped. So kind of like this crescent appearing structure, as opposed to the elongated, sort of striated nature that we see with skeletal and cardiac muscle. The length is approximately 1/10 the size of skeletal muscle. So they're much smaller cells than we see with other muscle types. They contain actin and myosin, so they do have the same filamentous proteins. They do have the same structural components, but they're just differently arranged so that they will behave a little bit more differently than what we see with those two muscles as well. There are no sarcomeres. So while there is actin and myosin, they are not arranged or organized in the sarcomere. And remember, the sarcomere is what gives the muscle fiber and eventually the entire muscle body its striate, kind of striped appearance. And this is why when you look at even if you think about skeletal muscle, looking at a cadaver, if you think about looking at meat, so chicken or fish, it has that striped, striated, muscular appearance because of the sarcomere. The sarcomere is what gives skeletal muscle and cardiac muscle its striped appearance. Because smooth muscle has no sarcomere, it is appearing smooth. There is the presence of a structure called dense bodies. And dense bodies are what help to anchor the actin and myosin. They help to anchor these structures together, and they're also going to be acting like the Z lines. So the Z lines were these point of anchoring. It was also the border between one sarcomere and another. And when the sarcomere shortened, the Z lines moved closer together. We're going to see a similar phenomenon. And so the dense bodies are going to behave in a similar way. They're going to anchor the filaments together. They're going to provide stability as the filaments slide past each other. And they're going to be shortening or coming closer together when the actin and myosin are sliding past each other, or when the cells are contracting. Okay, so looking at the sliding filament mechanism of contraction, which we would have talked about in skeletal muscle, and then pointing out here some ways that it is the same and some ways that it's different. So actin and myosin are also present, but they are longer. They are longer in smooth muscle, and so they allow for longer contraction. So there's more opportunity for sliding to happen as these things slide past each other because they're physically longer. We also see the arrangement is different. So they're not arranged parallel, in strips or stripes, the way that we saw them in the sarcomere. Because there's no sarcomere here, they're arranged in this oblique fashion, kind of running diagonally across the cell itself. The myosin head's whole length. So essentially, the myosin head is going to move all the way across the thin filament. So that's also going to give us a longer range of contraction. And we'll see that smooth muscle contractions are slower, they're longer, they're more sustained than the quick, rapid nature of skeletal muscle and even cardiac muscle contractions because of this nature. All right, so just to look here a little bit closer at what a smooth muscle cell looks like. So here's a smooth muscle cell. It's got the spindle-shaped arrangement or organization. It's got the oblique, the oblique laying actin and myosin, the orange squares in the middle being myosin, or the thick filament, if you will, the blue longer stripes being the actin or the thin filaments. And so as the thick and thin filaments slide past each other in a very, very similar way that we saw them slide or kind of drag past each other for skeletal muscle, they do that here. And as they do that, the entire cell kind of crumples up on itself. It kind of shrivels down as the dense bodies. So the dense bodies are going to be these central points here where we've got the thick filament doing the sliding. And so the dense bodies are going to kind of come down closer to each other, again, very similar to the Z lines coming closer to each other as the cell crumples down in its contracted state. All right, let's look at the steps of smooth muscle contraction. So we'll kind of list them out here, explain them, and then later on we'll illustrate them and talk about, how things are actually happening. Also talking about how they differ. So it's really important as we're going through this to pinpoint ways that they differ from skeletal muscle. This is how you're going to get your questions on the exam, and this is how you're going to be able to apply this information in a more meaningful way. So the steps of smooth muscle excitation, contraction, coupling start out with calcium, just like skeletal muscle did. But the calcium is now coming from a different source. So it can be coming from the sarcoplasmic reticulum to some extent, just like we saw with skeletal muscle, but then most of it also can come from outside of the cell. This extracellular source of calcium is a component here in smooth muscle contraction as well. We're going to see the opening up of voltage-gated calcium channels that are on the plasma membrane, also on the sarcoplasmic reticulum. That is going to trigger the release of calcium. So calcium is going to be moving from the SR, and it's going to bind to a structure called calmodulin. Together, calcium bound to calmodulin forms what's called the calcium calmodulin complex, and that complex together will activate MLCK, which is myosin light chain kinase, okay? Once MLCK is activated, that's actually an enzyme, hence that word kinase at the end there. That is going to phosphorylate myosin. So it's going to phosphorylate myosin. In other words, it's going to add that phosphate group to the myosin head. That is going to energize the myosin head, and that is going to allow for myosin to make interactions with actin and start the cross-bridge cycling. Beyond that point, cross-bridge cycling is the same event generally as it was in skeletal muscle, in that myosin will drag actin closer towards the dense bodies this time, and they're going to kind of shorten and then crumple down the entire cell, and that will generate a smooth muscle contraction. Okay. Notable differences are the fact that we got different players here. So we don't have, for example, troponin or tropomyosin regulating this process, but we still have calcium regulating this process as well. So calcium is still the regulator of this contraction. It's still turning it on and turning it off, but it's doing so by a different mechanism. Okay, so just to go through that here again, we've got calcium coming in from the external environment, along with calcium coming from the sarcoplasmic reticulum. Okay, so it's coming from both of these sources, and it's going to be dumped into the sarcoplasm, really the cytoplasm of the cell. From there, it's going to bind to calmodulin. So calcium binding to calmodulin creates the calcium calmodulin complex. And this complex together, which is basically kind of activating or turning on calmodulin. This complex, now that it's activated, will activate MLCK, so myosin light chain kinase, which is an enzyme. So once this enzyme is activated, it will then catalyze the reaction of phosphorylating the myosin head. So the myosin head, being the thick filament component of our muscle filaments, it needs to be energized. We made this, this point before. It needs to be energized in order to do the dragging of the actin filament. And so MLCK can phosphorylate it to help bring about that energized state so it can start the cross-bridge cycling. It can also work in the backward direction and unphosphorylate the myosin head to kind of turn it off and stop the cross-bridge cycling. So this can be going in forward or reverse directions by this enzyme MLCK. So if we have the unphosphorylated myosin head, we have no myosin ATPase activity, we have no cross-bridge cycling. we have no muscle contraction. If we do have the myosin head phosphorylated, we then see cross-bridge cycling, we then see muscle contraction. Okay, I do want to point out how we unphosphorylate the myosin head. So essentially, if there's no calcium present, so if the calcium is not here, if it didn't come in from the outside of the cell, if it didn't come in from the SR, then there's no calcium in the sarcoplasm, then we cannot turn on calmodulin and we cannot activate MLCK. And so that would result in unphosphorylation as well. So this is basically dependent on calcium in order to turn this off or to turn it on as well, right? In either direction. Okay. So let us look at this here in this illustration, and I will post this illustration with the drawings today, just in the event that anything's unclear. So don't feel too rushed or pressured to kind of copy everything down, but I will go through this. So it's more important, more valuable for you to kind of follow as I'm going through this. Okay. So we're starting out with our smooth muscle cell. We've got this calcium pump on the membrane here, the plasma membrane, that is the calcium pump on the sarcoplasmic reticulum membrane as well. Now, calcium comes into the cell when the membrane is depolarized. So if you think about the neuroeffector junction, remember the neuroeffector junction is where we have the postganglionic neuron synapsing onto a smooth muscle cell such as this. That depolarization, that stimulation of the muscle is what opens up the calcium channels. So we open up this calcium channel on the membrane. When the membrane depolarizes, we then trigger, once calcium starts coming in, we trigger the release of more calcium from the SR, from the sarcoplasmic reticulum here. This phenomenon is known as calcium-induced calcium release. So when calcium first comes into the cell from the plasma, from the outside of the cell, it then further triggers more calcium coming from the storage area here inside the cell. All that calcium floods the sarcoplasm, binds to calmodulin, and creates the calcium-calmodulin complex. Okay. The calcium-calmodulin complex will then activate MLCK. So it's going to turn MLCK on, and MLCK can actually phosphorylate the myosin light chain kinase. All right. Now, if we don't have MLCK, kind of moving in the backward direction, we've got phosphatases. So phosphatases are basically covalent regulation of this enzyme. So MLCK not turned on generates phosphatases, which unphosphorylate the myosin head, and that'll result in no muscle contraction. So MLCK phosphorylates the myosin head, phosphate cycling happens, muscle contraction happens. If there's no calcium in the sarcoplasm, we deactivate MLCK, phosphatases take over, we unphosphorylate the myosin head, no cross-bridge cycling, and no muscle contraction. Right. So the main point I want to make here is that calcium is still the regulator of all of this, but it's just not involving tropomyosin, not involving troponin. Instead, it is binding to calmodulin to turn this on, and then unbinding from calmodulin to turn this off. Okay. Let's talk about the relaxation of smooth muscle. So smooth muscle relaxes in a similar way that skeletal muscle does. We've got to remove the phosphatase from the myosin head, and we've got to clear the calcium from the sarcoplasm in order to do that. So there are active pumps that are pumping calcium back into the SR. That's going to be the calcium ATPase pump. But then because we have a second source of calcium, calcium also coming from the outside of the cell, we can also exchange that sodium coming in for calcium going out of the cell as well. So we can have counter transport where we can use the energy that sodium's liberating as it comes into the cell and take that energy, so secondary active transport, take that energy and move calcium back outside into the extracellular environment. Another important thing to note is that the myosin ATPase contraction is slower. So we kind of made this point before. Smooth muscle contractions are much slower, much more sustained, 10 to 100 times so than what we saw for skeletal muscle, right? So if we saw, for example, the muscle contraction for skeletal muscle, let's say we had a muscle that could contract in 10 milliseconds, we would see that in smooth muscle, that muscle would take 100 to maybe 1,000 times, 100 to 1,000 milliseconds, excuse me, to bring about that same contraction. So much slower contractions. Okay, now the regulation here happens via the autonomic nervous system. So we've got the sympathetic division, we've got the parasympathetic division. These are ideas that we talked at length about in chapter 11. This can be an excitatory result on the smooth muscle. That is usually a contraction of that muscle. This can be an inhibitory result. That would be a relaxation of that muscle. It's going to be dependent upon the receptor type. So whether we have an alpha receptor, or alpha 1, alpha 2, beta 1, beta 2, beta 3, that will determine the exact type of response that we see on that specific muscle tissue, depending on the location as well. The neurotransmitters being released from the varicosity here. So at the neuroeffector junction, remember we're seeing varicosities and not the end-to-end or the neuron-to-neuron synapse that we see at other synapses. We're seeing varicosities here, which are present at the neuroeffector junction. And then we see the diffuse binding of the neurotransmitter to the receptor, so they kind of cross the junction, bind to their specific receptor. Remember, the neurotransmitters here are norepinephrine or epinephrine if it's the sympathetic division, and then it's going to be acetylcholine if it's the parasympathetic division. It will bind to its respective receptor, and then that will generate muscle contraction or response.